ازاي تنجح وتجيب مجموع وانت قاعد على الانترنت؟ اليوم السابع بيقدم اقوى مراجعات نهائية للثانوية العامة لنخبة من افضل المدرسين. اتفرج وذاكر وانجح وجيب مجموع بالفيديو على اليوم السابع.
اهلا بيكم ابنائنا وبناتنا طلاب وطالبات الثانويه العامه في المراجعه النهائيه على اليوم السابع او من اليوم السابع على قناه اليوم السابع وموقعه الالكتروني وعلى اليوتيوب. نبدا ان شاء الله الحلقه الثانيه من ماده البيولوجي. هنكمل باقي الريفيجن اللي احنا بديناه من الحلقه اللي فاتت. The question asked about give the scientific term for each of the following. Number one, part of the brain which all sensory impulse except the smell pass through before they reach to the sensory centers in the brain. The question asks about the part of the brain that's responsible for the coordination of the sensory centers except smelling. Of course, the answer is the thalamus. Thalamus is a part of the brain that is responsible for causing the coordination between the centers of the brain except the smelling, because it's not reached the thalamus and controlled by the brain cortex. The next question. A hormone affects and stimulates the physical and mental growth and development. The hormone responsible for the stimulation or the stimulates the physical and the mental growth and its development. The question asked about the thyroxine hormone that's responsible for the development of the mental and the physical growth of the human being. The next question. A hormone secreted by pituitary gland increases the blood pressure. What about the hormones that's responsible for increasing the blood pressure? This hormone is secreted by the hypothalamus, which acts as a part of the forebrain. This hormone has two names. The first is the antidiuretic hormone. The second is the vasopressin. The vasopressin is a hormone which is secreted from the hypothalamus and passing through the posterior lobe of the pituitary gland and responsible for increasing the blood pressure and decreasing the amount of urine. The next question. A flat bone pointed at the lower part which made of cartilage. This bone of the thoracic cage that connected with the upper 10 pairs of ribs only, and the lower part is called the cartilage. What about the name of this bone? Of course, that's called the sternum bone. The next question. The point of the connection between terminal branch of the nerve fiber and the motor in the plate of the muscle fiber. The question asked about the point of connection between the terminal branch of the motor fiber and the motor plate. Of course, if this part is the arborization of the motor nerve and that's the plate of the muscle, this part is called the neurojunction muscle. So that's the definition of the neurojunction muscle. The next question. The cells which secrete fluid nourish the sperms inside the testes. The question asked about the cells inside the testes that are responsible for the secretion of substance that to feed the sperm. Of course, these cells is the Sertoli cells. Sertoli cells responsible for the secretion, and also it has an immunization function. The next question. They also ask about the cells inside the testes that believed to have an immunization function. Of course, this function is the immunization function of the sperms. So that's the Sertoli cells also. The next question. The ability of unfertilized egg to develop into a new individual. The ability of the unfertilized egg to develop to produce complete individual. That means the egg produce complete individual without fertilization. Of course, that takes place naturally in the honeybee and the aphid insect. That's called the parthenogenesis. The next question. The transmitters of plasmodium among the human being. What about the living organisms that are responsible for the transmission of the plasmodium of malaria to the human being? Of course, that's the infected female Anopheles mosquito. The infected female Anopheles mosquito can transfer or carry the stages of the plasmodium malaria, which reproduce sexually inside the female Anopheles mosquito, and after the certain kind of reproduction sexually and asexually inside the female, the sporozoites get out from the salivary glands of the female and infect the person. The next question. The human organ where sporozoites spend the incubation period. The question asks about the sporozoites which get inside the human being. What about the organ where it located inside this organ and divided to produce a great number of merozoites? Of course, the answer is the liver. The liver is the organ where the sporozoites spend the incubation period. The next question. The phase through which the spermatids are converted into sperms. The spermatids exchange into sperms in the metamorphosis stage. So the question asks about the metamorphosis, which acts as the last stage in the spermatogenesis. The next question. The phase when the ovary stimulated to produce a Gravian follicle. The phase when the ovary is stimulated to produce a Gravian follicle. Of course, that asks about the phase where the pituitary gland secrete FSH hormone or follicle stimulating hormone, which stimulates the ovary to form the Gravian follicle. That's called the proliferation stage. Because the Gravian follicles create estrogen hormone, which is responsible for increasing the thickness of the endometrium and the regulation of the menstruation cycle of the human female. The next question. The fusion of male nucleus with the two embryo sac nuclei to form endosperm. The question asks about fusion of male nucleus and the two embryo sac nuclei. That means that will form 3N, which is called endosperm. Therefore, that's act as a very simple question because the answer is a triple fusion. Triple fusion means a fusion between the male nucleus and the two embryo sac nuclei of the embryo sac of the flower to form the endosperm. The next question. This part explains that two generative nucleus. One of them is fused with the egg cell, this egg cell to form zygote, and the other generative nucleus combined with the two embryo sac nuclei to form endosperm. So that's called the triple fusion. The value and benefit of any experiments performed is determined by the degree of the adaptation of the tools and the materials to the experiment. Of course, that's in genetics. Mendel states that the adaptability is the value and the benefit of any experiments performed is determined by the degree of the adaptation of the tools and the materials to the experiment. The next question. The exchange of pieces between the two inner chromatids of the homologous chromosomes in the prophase 1 of the first meiotic division. During the meiotic division or the first meiotic division during prophase 1, tetrad is formed. Tetrad means crossing over takes place between the two homologous or two inner crossing over chromatids, and the exchange that takes place, this exchange is called the crossing over. So crossing over is responsible for the formation of new genetic traits of inside the cell. The next part or the next question. A group of nitrogenous bases has single ring. A group of nitrogenous bases has single ring. We understand that the nitrogenous bases are divided into two main groups. Purine nitrogenous bases consists of two rings as adenine and guanine. The pyrimidine is a single or as kind of nitrogenous base consists of single turn, so the single ring that's called as the T and the C or thymine and cytosine. The question asks about the nitrogen species with single ring, which is called the pyrimidine, as thymine and the cytosine. The next question. Enzyme used to build up DNA, it's called present in the virus with the RNA genome. The virus which containing RNA as a genome or as a genetic material needs the presence of certain enzymes to change the RNA into DNA to combine with the other cells. Therefore, the enzyme is called the reverse transcriptase enzyme, because we understand that the transcription is the formation of RNA from DNA, but reverse transcription changes RNA into DNA, so the enzyme is called the reverse transcriptase enzyme. The reverse transcriptase enzyme is the enzyme in virus which containing the RNA, the genetic material. The next question gives scientific term also. mRNA molecules which has several to over 100 ribosomes attached to it. The question asks about mRNA containing more than 100 ribosomes. Therefore, that called polyribosomes or polysome. So the answer is a polysome or polyribosome. That accelerates the rate of the production of the protein cells. An enzyme follows the helicase enzyme directly during DNA duplication. We understand that the DNA is duplicated by the presence of three enzymes: helicase, DNA polymerase, and ligase enzymes. The two enzymes are move in one direction is the helicase and the DNA polymerase. So the question asks about the DNA polymerase, which form one of the two strands of DNA and add nucleotides to the three ends to allow to help the ligase enzyme to join the nucleotides to form the other strand. The next question. A phenomenon in which the number of chromosomes are duplicated during the gamete formation of meiosis or during mitosis. Of course, the phenomenon is the polyploidy. That means change 2n into 4n, 6n, 8n, and so on. That due to the absence of middle lamella that act as the induced mutation. So that's the polyploidy. The next question. The conversion of mRNA codon into a particular sequence of amino acids. The conversion of mRNA codon into a particular sequence of amino acids. Of course, the answer is the translation or the protein sense. The translation is the translation of the codon mRNA into amino acids and joining the amino acids to form the polypeptide chain that takes place by the ribosome. The next question. A protein substance decreases the ability of viral genetic material to duplicate. That means the question asks about a certain kind of protein that interferes the duplication of the viral genetic material and duplicates the neighbor cells from the infection of the virus. Of course, the question asks about the interferon. The interferon is a protein substance that decreases the ability of viral genetic material to duplicate and to protect the cells against the infection of virus. The next question. An enzyme snaps the DNA at or near the recognition site. A certain sites are located on the DNA molecule, but these enzymes that are responsible for snips or cut DNA with two inclined sticky ends. Of course, that's a very simple question because the answer is the restriction enzyme or restriction in the nuclease enzyme. The next question. The next question is a certain kind of question which is called the give reason for each of the following. The give reason question, that's the question, the first question which asks about HCl. It play an important role for the digestion in the stomach. The question asks about what's the importance of HCl in the stomach. Of course, HCl responsible for number 1, kill the microbes. Number 2, activate the pepsinogen into pepsin. Number 3, that keep the medium strong acidic to help the pepsin to act. So the answer, number 1, it killed the microbes which entered into the stomach with the food. Number 2, it activates the pepsinogen into pepsin. Number 3, it makes the stomach medium acidic 1.5 to 2.5 to help the pepsin enzyme to act, which change the protein into polypeptide. The next question. The next question give reason, ask about a certain part of the photosynthesis process. The complete process of photosynthesis can take place in the dark. The question asks about why the photosynthesis not takes place in darkness only. But the question asks about what about the importance of dark reaction or importance of light reaction. So the answer, during the light reactions, net pH2 is formed and the ATP, which are used to reduce carbon dioxide to form sugar. So the answer, because net pH2 and the ATP are produced from the light reaction to reduce carbon dioxide into carbohydrate into dark reaction. So dark reaction can't take place at the absence of net pH2 and ATP, which are formed as a result of light reaction. The next give reason, which ask about the blood inside the blood vessels. The blood never clot inside the blood vessels. Why the blood is not clots inside the blood vessels? That because of 3 reasons. The first reason, the blood runs inside the blood vessels in a regular speed and doesn't slow down. Number 2, the blood platelets slide easily smoothly inside the blood vessels in order not to be broken. Number C, the liver secretes heparin substance that prevents the activation of prothrombin into thrombin. The next question. The next question asks about the blood cells or the red blood cells. The body benefits from the dead red blood cells. How the body can benefit from the dead red blood cells? Of course, that's a simple question, because the liver used the hemoglobin of dead red blood cells to form bile, and we understand that the bile responsible for the change fats into emulsified fats by decrease its surface tension or increase the surface area that exposed to lipase enzyme. So the body uses the hemoglobin of dead red blood cells to form the bile that takes place inside the liver. The next give reason question was to ask about the sounds of the heart. 2 sounds of the heart beats can be distinguished by the stethoscope of the doctors. If you hear the sound of the heart, that's called doblop, doblop, doblop. So what about these 2 sounds of the heart? Of course, the first sound, which is called the lub sound, that's act as a long low pitched, that due to the closure of the bicuspid and the tricuspid valves between the atria and the ventricles. The sound of the 2 valves that takes place due to the closure of the bicuspid and the tricuspid valves during the contraction of the ventricles. But the dub sound is a short high-pitched sound that due to the closure of semilunar valves in pulmonary artery and the aorta artery. So due to the closure of semilunar valves during the relaxation of the ventricles or contraction of the auricles. The next question give reason. A Krebs cycle doesn't require the presence of oxygen and can't start as the absence of oxygen. Krebs cycle doesn't require the presence of oxygen and can't start as the absence of oxygen. 2 questions. Why the Krebs cycle doesn't need the presence of oxygen? And the second question, why it can't start as the absence of oxygen? Of course, we understand that the Krebs cycle doesn't need the presence of oxygen because the oxidation takes place by losing electrons and the protons that carried on NAD positive and FAD, or NAD H and FAD H2, and the oxidation is not takes place by the oxygen but by losing electrons and the protons carried on NAD and FAD. But why it can't start as the absence of oxygen? If the Krebs cycle doesn't need the presence of oxygen, why it can't start as the absence of oxygen? Because we remember that at the absence of oxygen, pyruvic acid is reduced into ethyl alcohol and carbon dioxide or lactic acid according to the cell. But during the absence of oxygen, the pyruvic acid can't oxidize into acetyl group, so it can't get in Krebs cycle as the absence of oxygen, because need the oxygen to oxidize the pyruvic acid into acetyl group to get in Krebs cycle. But the absence of oxygen, the pyruvic acid is reduced into lactic acid or ethyl alcohol and carbon dioxide, that according to the cell. The next question, give reason. The next question asks about the sweat gland. The sweating increase in summer. The question asks why sweating increase in summer. Of course, in summer the temperature increase, so dilation takes place of the blood capillaries in the skin. That means a great amount of blood reaches the sweat gland, that activates the sweat gland to absorb or extract a great amount of the sweat from the blood, that get out through the sweat pores, that causing the evaporation of the sweat from the skin, that decrease the body temperature. So in summer the temperature increase, so the blood in the blood capillaries surrounding the sweat glands increase, that increase the activities of the sweat gland to extract the sweat components from the blood through the sweat ducts to pour to decrease the body temperature in summer. The next question, give reasons the presence of enough amount of cholinesterase enzyme at the neurojunction muscle. The question asks about what's the function of the cholinesterase enzyme. We understand that the cholinesterase enzyme responsible for breaking down the acetylcholine after performing its function, because when the acetylcholine passing through the cleft and reaches the muscle plate, it changes the state of polarization into depolarization, that causing the contraction of the muscle. So we need the muscle to relax or change from polarization into repolarization. That means we need to breaking down the acetylcholine, which act as a stimulus of the membrane of the muscle. Therefore, the cholinesterase enzyme that needed to destroy the acetylcholine into choline and acetate after performing its function, and then the membrane of the muscle fiber returns its polarization. That means the resting state, the muscle fiber membrane is ready to be stimulated at the response again. So the cholinesterase enzyme breaking down the acetylcholine into choline and acetate after performing its function to change the muscle plate to the state of polarization or repolarization to start to contract again. The next question. The next question asks about, give reason about the blood groups. The blood group AB, or the person of the blood group AB, can considered as a universal recipient. Of course, the blood group AB, that means on the surface of RBC the presence of antibody A and antigen A and antigen B, that's on the surface of the red blood cells. But in the plasma of the person which containing the blood AB, there is no the presence of antibodies. So that means the person which containing or which has the blood group AB act as a universal recipient because it has no any antibodies against the other blood groups. So we can transfuse the blood from any blood group to the person of AB. But also the blood group O act as a universal donor because the blood group O, that means it has no any kind of antigen but containing the antibodies A and B. The next question, virus can grow inside a certain kind of bacteria while they can't grow inside the other strains. The virus can grow inside a certain kind of bacteria as the certain strains of E. coli bacteria, but can't survive on the other kind or other strain which containing the resistance against the virus. Of course, the answer related to the presence of certain enzyme which cuts the DNA into pieces with two inclined stick ends. Of course, the enzyme is a restriction in the nuclease enzyme. So the answer, because the resistance bacteria produce restriction enzymes that attach specific nucleotide sequence in the viral DNA, breaking it into useless fragments. The next question, give reason question. The ability of the bacteria to degrade the viral DNA. The ability of the bacteria to degrade the viral DNA. Of course, that because these kinds of bacteria containing the kind of enzyme which is called restriction endonuclease enzymes that cuts the DNA into useless pieces so that protect the cell against the infection with the virus. But these cells which containing the restriction enzyme containing a modification enzyme which put methyl group on the recognition sites that protect the DNA of the bacteria against the action of the restriction enzyme. The presence of the reverse transcriptase in the virus whose genome is RNA. The reverse transcriptase used by the virus that to change the RNA into DNA, that to combine with the genome of the bacteria or of the host's DNA. That means the virus needs the presence of reverse transcriptase enzyme to reverse transcription or change the RNA, which is the virus genetic material, into DNA, that to help the virus to join with the host's DNA to causing the infection. The next question is a certain new kind of questions. It's mention site and the function of each of the following. The mesenteric membrane, of course, the site. We study the presence of mesenteric membrane between the coils of the small intestine that to feed, support the small intestine and protect it against the coiled and connected together. So the site between coils and the loops of the small intestine, the function connects coils and the loops of the small intestine together. The next question mentions site and the function. Lacteal vessels. Lacteal vessels. Of course, the lacteal vessels is a part of the lymphatic system and located also inside the villi, that to absorb the vitamins that dissolved in fats as a KDA vitamins, fatty acid and glycerol, to carry them to the bloodstream to join the blood with the superior vena cava of the heart. So the lacteal vessel site is connect the lymphatic vessels node and the nodes of the lymphatic vessels. It's also located in the villi and located in the many parts of the body, organs and body tissue. They collect the vitamins K, D, A and A with the fatty acids and glycerol to the lymphatic vessel and follow it in the right atrium of the heart through the superior vena cava. The next question, which asks about the glutation or the hydrosol. The hydrosol also is called water stoma. The water stoma is a group of loose cells, one or many cells are loose that to allow the passage of water which containing salts to get out through it, to get out from the leaf through the growing tips of the leaf. That means it containing water and salts, because during the evaporation of the water which containing salts we can observe the presence of white powder on the growing tip after the evaporation of the water. That means the water of glutation containing the kinds of salts that act as a kind of excretion implant. So the site of hydrosol, one or many loose cells as the tips of some plant leaves as a potato and a tomato. But the function through which the glutation occurs. Glutation means getting rid of water and salts as a kind of excretion in some plants that takes place during the early morning during the spring. The next question mentions the site and the function of each of the following. Synaptic vesicles. Inside this knob, the presence of vesicles which containing neurochemical transmitters. The neurochemical transmitters are responsible for the transmission of nerve impulses through synapses, because we understand that the transmission of the nerve impulses through the nervous tissue that takes place by or through the nerve fiber, and transmission of nerve impulses through the synapse. The synaptic vesicles is located inside the synaptic bottom. This bottom is the synaptic bottom, where the presence of these vesicles which containing neurochemical transmitters. The function filled with the chemical transmitters which play an important role in the transmission of the nerve impulses from one neuron to the next one. The synaptic vesicles can carry the neurochemical transmitters as acetylcholine and noradrenaline to allow causing the transmission of the nerve impulses from one neuron to the next one, from neuron to the muscle, or from neuron to the gland, which is the kind of synapses, where the presence of the neurochemical transmitters which act as a chemical substance that transmits the impulses from one neuron to the next, or from one neuron to gland or muscle. The next question mentions the site and the function of the acrosome. We remember that the acrosome site in the head of the sperm. We remember that the head of the sperm containing a nucleus and the acrosome. The nucleus that containing the genetic materials that transfer from the father to the offspring, or from the male to the offspring, and also containing the acrosome. We understand that the acrosome responsible for the secretion of hyaluronase enzyme that dissolves the hyaluronic acid to remove the coat around the ovum to facilitate the penetration of the sperm through the ovum to facilitate its fertilization. So the site in the head of the sperm, the function, it secretes the hyaluronase enzyme that dissolves a part of the ovum membrane to facilitate its penetration process, or to facilitate the fertilization, because the coat that located around the ovum is attached to the ovum by the hyaluronic acid, and the hyaluronase enzyme dissolves the hyaluronic acid. So we need the presence of great number of sperms to produce great amount of hyaluronase enzyme to remove the coat around the ovum. The next question mentions the site and the function of each of the following. The prostate gland. The prostate gland is located in the human male reproductive system, around the neck of urinary bladder. The prostate gland is responsible for the secretion of a nourish substance to feed the sperms, and also secrete alkaline substance to facilitate the or to neutralize the medium of the genital or urinogenital duct, that to facilitate the passage of the sperms, because the acidity can kill the sperms. So we need to causing neutralization of the genital duct to facilitate or to help the sperms to pass through it. So the site is the male reproductive system around the neck of the urinary bladder. The function, they secrete the sugary fluid to feed the sperms, and secrete the alkaline fluid to neutralize the acidity of the urethra just before the sperms. Urethra means the urinogenital duct, that because the acidity acts as a harmful medium of the sperms, so we need to neutralize the medium that takes place by the alkaline substance that's secreted by the prostate gland. The next question mentions the site and the function of the corpus luteum. We understand that the hormone which is called the luteinizing hormone that's secreted from the pituitary gland, or the anterior lobe of the pituitary gland, which is called the adenohypophysis. The adenohypophysis part which secretes the LH hormone that stimulates the gravium follicle to release the mature ova and change into corpus luteum. But the corpus luteum responsible for the secretion of estrogen and the progesterone that increase the thickness of the endometrium and adhere the endometrium in the uterine muscles. So the site is inside the female ovary. The function, the production of the mature ova. Number 2, the secretion of female sex hormones that responsible for the increasing the thickness of the endometrium and the vascularity of the endometrium during the menstruation cycle. The next question, what would happen in each of the following cases? What would happen in each of the following cases? Number 1, introducing a piece of bread inside the mouth and chewing it continuously for 3 minutes. If you get a piece of bread inside the mouth and chewing it for about 3 minutes, what would happen of this piece of starch or piece of bread? We understand that the bread containing starch or made of starch, and inside the mouth the salivary gland secretes saliva which containing mucus and containing ptyalin enzyme. The ptyalin hydrolyzes starch into maltose, so it becomes sugary more sweat, so the starch change into maltose sugar by the effect of thiolane enzyme, because the starch act as a polysaccharide, but the maltose act as a disaccharide that takes place by the action of thiolane or the salival amylase inside the mouse, which act in a very weak alkaline medium. The next, what would happen question, which ask about the heart. If you cut, what would happen if you cut the sympathetic nerve and the vagus nerve from the sinoatrial node? We understand that the sinoatrial node is connected with the two nerves, vagus and sympathetic nerve. The vagus nerve inhibits the heart beating, sympathetic accelerate the heart beating. So the vagus, if we cut the two nerves, that means the regulation of the heart beating or the rate of the heart beating will not change according to the various stimulation, because the two nerves will not act. So the heart beating takes place by the sinoatrial node, which is not affected by the other conditions. أعزائي طلبات الثانوية العامة، دي كانت نهاية الحلقة الثانية من مادة البيولوجي من اليوم السابع. نتمنى أنكم تكونوا استفدتوا منها كلكم إن شاء الله.
إزاي تنجح وتجيب مجموع وأنت قاعد على الإنترنت؟ اليوم السابع بيقدم أقوى مراجعات نهائية للثانوية العامة لنخبة من أفضل المدرسين. اتفرج وذاكر وانجح وجيب مجموع بالفيديو على اليوم السابع.